Well, good afternoon, everyone. So far we already finished the citric acid cycle and also the glycolysis. So these two important metabolic pathways to generate ATP, right? So we know there are some differences between these two metabolic pathways, especially for the citric acid cycle. It is a hub that connects the glycolysis and the later one, so which we're going to discuss today, the oxidative phosphorylation. They have something in common that they all generate ATP, the energy. And there are some differences also, right? Like aerobic with oxygen or without oxygen. So the one we're going to discuss from today, the oxidative phosphorylation, you can tell from the name, oxidative phosphorylation, right? So it must be in the aerobic condition with oxygen. So before we talk about oxidative phosphorylation, we're going to study some contents from chapter 13, which is talking about the membranes. Share. So for chapter 13, you know how to start the whole chapter, but only the introduction and the 13.1 section. And if you don't know how to start, you just focus on the notes I wrote. So only these parts you need to know. Basically introduce some concept about the membranes or related with the membranes. The lipid bilayers of biological membrane is intrinsically impermeable. Basically, we see the inner membrane of the cell, the mitochondrial cell, is impermeable, which means it cannot let the ions and some polar molecules pass through. However, this species, some of these ions or some molecules must be able to cross this membrane for some normal function of the cell. So then what can the cell do? There must be some ways the cell can do to make sure these molecules can cross the membrane. So the permeability is conferred by three classes of membrane proteins. That is what you need to know. There are pumps, carriers, and ion channels. So for pumps, basically they use the source of free energy, for example, ATP, hydrolysis, or the light absorption. They use the energy from these reactions to overcome the high activation energy of ions or polar molecules, or just overcome this energy to make these ions or molecules pass through the membrane. So this actually is an example of active transport. So here, when we say high activation energy, sometimes you can also say drive up here energy. Okay? So this is an example of active transport. So later on, we're going to explain what is active transport. Actually, is the primary active transport. So carriers will mediate the transport of ions and molecules across the membrane without using ATP, but using the gradient of one ion to drive the transport of another ion or molecule. So you won't use energy from ATP, but it will, use the gradient. So later on, we will explain how can this gradient be generated, right? So overall, it comes from different concentrations of the proton. But this gradient of one ion will drive the transport of another ion or molecules, like a series effect, right? So then this is an example of secondary active transport. Both active transport a little bit different. Use ATP or use gradient. So the third part is the ion channels. We're talking about pumps. We're talking about carriers. Now we talk about ion channels. The ion channels will provide a membrane pore through which these ions can flow rapidly in a thermodynamically downhill direction. So which means this is a channel, right? So there are some pores. So through these pores, the ions can flow. So flow from a higher energy side to a lower energy side. So that is downhill direction mean from high energy to lower energy. And these ion channels are example of passive transport, or it's called facilitated diffusion. So now we introduce the concept of passive transport and active transport. Passive transport is a method of ions and molecules to cross the membrane. In this type of transport, the energy driving the ion move across the membrane originate from the ion gradient itself and is a passive transport. And so, in another way, the active transport, it will require an input of energy for another from another source to let this ion and molecules cross the membrane, which means you do need to input some energy. But for passive, you don't need to input energy. Instead, you only need to use the gradient, right? So the gradient itself. So those are the three comparts for the membrane and the two transport method we have to understand. Okay. Later on, when we started the oxidative phosphorylation, so for some steps, some reactions, you need to differentiate is active transport or is a passive transport. We will see that. We will come back to the definitions when we get there. So now, here is another question. So the amount of ATP for the human being required is a large amount. So one day we require 8,400 kilojoules, which means we need 83 kilograms of ATP. But the human being possess only 250 grams. That's far away from what you need, right? 250 grams compared with 83 kilograms, like more than 100. I mean, more than 1,000 far away. So how can we meet this requirement? The only way is you have to recycle the ADP back to ATP, because when you hydrolyze ATP, you form ADP. If we can use this ADP to synthesize ATP again, then we can generate more energy. This actually can be done in a very short time, right? So each ATP molecule is recycled 300 times per day. In that way, our body can provide enough ATP for us to do the work or to do exercise to help the cells function. And this recycling takes place primarily through oxidative phosphorylation. That's why this oxidative phosphorylation is very important, because we know, okay, glycolysis can generate ATP, the citric acid can generate ATP, but the amount is really too small, not enough at all, right? So you have to generate more ATP through a more effective way. That is this oxidative phosphorylation. So this metabolic pathway actually is a series of oxidation reduction reactions, which allow the flow of electrons from NADH and FADH2 to oxygen. Or in another way, we say during this oxidative phosphorylation, you involve NADH and FADH2, which will be oxidized by oxygen. And of course, oxygen will be reduced. So oxygen is your oxidizing agent, and this NADH, FADH2 will be your reducing agent. So now you say, before we always say NADH, FADH2, they are electron carriers. Now we know where are those electrons will go. Eventually, they will be accepted by the oxygen. So those electron flow will take place in 4 large protein complexes, which we will talk details in, I think, section 5. Yeah, section 5 and section 6. So this complex, these 4 large protein complexes are embedded in the inner mitochondrial membrane, which means this oxidative phosphorylation occurs in the inner mitochondrial membrane. And this whole process, these 4 large protein complex, they are called respiratory chain or the electron transport chain. So here I forgot to write, it should be ETC. When you say this ETC, it means electron transport chain. Okay. And a general reaction for the NADH react with oxygen is shown here. So you have NADH act with oxygen. So NADH will be oxidized to NAD+, and oxygen will be reduced to water. So the delta G for this reaction is negative 220, which is very negative. So which means this is a spontaneous process. And even more, 3 of these complexes of the electron transport chain will use energy released by the electron flow to pump hydrogen plus out of the mitochondrial matrix. And the resulting unequal distribution of this hydrogen plus will generate a pH gradient and then a transmembrane electrical potential that creates a hydrogen motive force. So that we will see this later. Okay? Again, we will see this later. So the ATP synthesis with the hydrogen plus flow back to the mitochondria. ATP is synthesized from ADP and catalyzed by an enzyme complex, which is ATP synthesis. Here is the reaction. So basically, this ETC, the electron transport chain, is coupled with the ATP synthesized. Always say, in order to synthesize ATP from ADP, you have to make sure the electron transport chain works well. And you can tell this delta G is positive 30.5. So still remember that the hydrolysis of ATP is a spontaneous process with delta G negative. So the reverse reaction ADP from ATP is having a positive 30.5. So you have to couple these two reactions to make the overall process become spontaneous. Okay. So that's confirmed the importance for the ETC. So overall, the generation of high transfer potential electrons from TCA cycle will flow through the respiratory chain accompanied by the synthesis of ATP. So basically we keep seeing TCA will not generate many ATPs. Okay, only one ATP is directly generated. However, we keep seeing it will generate high electron carriers, high potential of electron carriers. That is NADH and FADH2. So now we're going to say, how can the NADH and FADH2 generate ATP? So again, so basically these are a big picture of the ETC coupled with ATP synthesis, or we say how ATP is regenerated through ADP, and why we have to know what happened for the oxidative phosphorylation, or we say the electron transport chain, right? So now we still start from talking about the membrane system. So we know, okay, oxidative phosphorylation, it occurs in the inner membrane, inner mitochondrial membrane. So still remember where the TCA cycle occur? In the matrix of mitochondria, right? So here the oxidative phosphorylation occurred in the inner membrane of mitochondria. The mitochondria actually has two membrane systems, the outer and the inner. The outer membrane is very permeable. Many RNA molecules can pass through. However, the inner membrane is very impermeable. So that many RNA molecules cannot pass through. And even more, the inner membrane folded into a lot of internal ridges, folded layers, right? That's called crystic. So there are two compartments in the mitochondria, inner membrane space and the matrix. We already know matrix TCA cycle occurred here, takes place here. And the intermembrane space is a space between inner and outer membrane. The inner membrane is where the oxidative phosphorylation occurs. So basically, both the TCA and the oxidative phosphorylation occurred in the mitochondria. By the way, there's a figure 18.2 which indicates the structure of the membrane. You can check your textbook or the PowerPoint. So since is impermeable, and we just learned in chapter 13, right? There are pumps, carriers, and ion channels which will help the molecules to pass through. And the pathway or the metabolic pathway that help these ions or molecules pass through is electron transport chain. So again, a big picture. So let's say if the oxidation reaction starts from a glucose. We know glucose, initially you should have the glycolysis and then pyruvate. Further, their citric acid cycle and then their oxidative phosphorylation. So after a series of reactions, eventually the glucose will be oxidized to be carbon dioxide and oxygen will be reduced as water. So this process will generate 10NADH. This is a NADH, 2 FADH2, and 4 net ATP. So why is this? That is the part you need to review if you get confused. Because I'll wait here. Okay, maybe we can talk a little bit later. But keep in mind, it will generate this amount of NADH, FADH2, and ATP. And those NADH, FADH2 are the cofactors that transfer electrons between glycolysis, TCA, and the protein complexes when you have electron transport chain. Okay? And overall, I think now we can check why you have 10 NADH is generated. That seems a lot. You don't remember we have so many NADH. But it seems yes. Why we have this many? So if you go back, check the process for glycolysis. There should be totally 2 NADH generated from glycolysis. Okay, you go back, check the 10 steps, which I won't show here. But there are 2 NADH generated. And also we know from the glycolysis, there are 2 pyruvate are generated, right? Because 3 carbon is converted from 6 carbon, so 1 glucose will generate 2 pyruvate. And each pyruvate will convert to acetyl-CoA and to generate 1 NADH. Make sure. Okay. So when you have pyruvate convert to acetyl-CoA, each pyruvate will generate one NADH. But remember, we have two pyruvate from one glucose. Right? So overall, you should have two NADH. And when this pyruvate is converted to acetyl-CoA, again, you got two pyruvates, right? Each acetyl-CoA will enter a citric acid cycle. So you have two acetyl-CoA. You're going to enter two rounds of citric acid cycle. And each citric acid cycle will generate 3 NADH. So overall, you will generate 6 NADH. Each round will generate 3 NADH. So 2 rounds will generate 6 NADH. Again, this is from one glucose. Okay, this is from one glucose. So everything for citric acid cycle should times 2. And everything from pyruvate should also times 2. Because each glucose will generate 2 pyruvates. Right? So that is how we get this 10 NADH. And the 2-FADH2 is easier to understand because each citric acid cycle will generate 1 FADH2. So you have 2 rounds of TCA from one glucose. So then you have 2 FADH2. It's 4 ATP. So one glycolysis, one glucose will have one glycolysis, which will generate 2 ATP. And then each TCA will generate 1, but you have 2 rounds. So you have 2 ATP. All right, so that's how these numbers comes from. Again, it means for one glucose. So now let's say how these two factors will generate ATP. So one NADH from the cytosol. If you see the C in your PowerPoint, it means cytosol, will generate 1.5 ATP. One NADH in the mitochondria will generate 2.5 ATP. One FADH2 will generate 1.5 ATP. So here also in the mitochondria. Okay, so be able to differentiate when NADH is located in different position of the cell, it will generate different amount of ATP. Okay, so that's how to be careful. So now let's calculate how many ATPs can be generated from one glucose. So when you have this 10 NADH, 2 NADH is generated from glycolysis, and we know glycolysis occurs in the cytosol, right? So these 2 NADH, each one will generate 1.5 ATP, NADHC, right? Because cytosol only 1.5 ATP. So totally you should generate 3. But this pyruvate converted to acetyl-CoA, that occurred in the mitochondria already. So that is 2 times 2.5. Each NADH in the mitochondria will generate 2.5. And the citric acid cycle also in the mitochondria. So it's 6 times 2.5. So FADH2, 1.5, 2 times 1.5. So ATP, they are the same number, right? So when you add all this together, so totally you actually got 30 ATPs. So that means from the oxidation of one glucose, you can maximum get 30 ATPs. So I can change this question a little bit. For example, I don't ask you how many ATP will be generated from one glucose. If I ask you how many ATP will be generated from one pyruvate, then you don't need to worry about this. And you do need to consider this pyruvate from citric acid cycle, but you only consider one round, right? So that will be, okay, 1 NADH from pyruvate and 3 NADH from citric acid cycle. So totally you should have 4 NADH from 1 pyruvate. And then you're going to have 1 FADH2 because only one round. And then no these 2 ATP, right? So only this 1 ATP from one round of TCA cycle. So overall, you will have 1 ATP, you will have 1 plus 3, 4 NADH, and 1 FADH2. So you can calculate how many ATP. Okay, so that is how to calculate the ATPs from different pathways. So now, when we talk about the oxidative phosphorylation, we have to consider a lot of the energies, and even more, we need to consider another term related with the energy, that is the cell potential. If you still remember in Gen Chem 2, right? So the last section we discussed about cell potential. So basically the cell potential, you have to consider two half reactions, a half oxidation reaction and a half reduction reaction. That means for the cell potential, you have to consider electron transfers. And that is exactly what happened to the oxidative phosphorylation. You have electron transferred. Okay. So just a quick review about the cell potential. And there's the relationship between cell potential and the Gibbs free energy. So we use the Gibbs free energy to measure the phosphoryl transfer potential. Remember in chapter 15? So we consider this delta G, right? Basically, we already know when delta G is negative, the reaction is spontaneous, which means the more negative the delta G, the higher the potential it will be. So now, when we talk about electron transfer, so we use the cell potential to measure the electron transfer potential. So how easy this electron can be transferred, right? So the more positive delta E, the higher electron transfer potential it will be. Or we say the higher the electron affinity, the higher the electron affinity it will have. So it will be easier to be an electron acceptor. In another way, so when we say delta G negative, reaction is spontaneous. So here you should have opposite way. When the cell potential positive, the reaction is spontaneous. Okay, so how to differentiate that? And there is a positive equation to describe the relationship between delta G and the cell potential. That is here. So the delta G equal to negative N times F times E. So the N is how many electrons are transferred. The F is a constant called Faraday's constant. And the E is the cell potential. Okay, so you notice the negative sign here. And that's why when delta E positive is spontaneous. Okay. So there is a table in the 18.1. It shows you the cell potential for some of the cofactors some of the molecules. Here, it shows you the cell potential values, and this N again is how many electrons are transferred. Okay, and if you notice, this table is from negative value to be more positive value. And remember, we define the hydrogen plus gain the 2 electrons become hydrogen gas. That is a SHE, right? Standard hydrogen electrons. So above is negative value, below is positive value. So the more negative the cell potential is, the more negative the cell potential is, the easier it will donate electrons. The more positive the cell potential is, the much easier it will accept electrons. So you can see oxygen in this table. Oxygen has the highest cell potential. So it's the molecule that's easiest to accept electrons. Here. Another thing you need to know here. So the negative reduction potential, like I showed in the table 18.1. So it means that this oxidized form of a substance has lower affinity for electrons compared to the standard hydrogen. So what does that mean? For example, let's say the table find NADH. NADH, NAD plus, right, is here. It's negative 0.32, 0.32. So that means the oxidized form, so the NAD plus, it has a lower affinity to electrons compared to hydrogen. Or we say when NAD plus react with hydrogen, so the NAD plus should donate electrons, right? Because hydrogen should have a higher affinity to electrons. So NAD plus will donate electrons to hydrogen. Okay. Yeah. So if it has a positive reduction potential, that means the oxidized form of the substance has a higher affinity for electrons than hydrogen. For example, oxygen. It has a positive 0.82. So that means when oxygen reacts with hydrogen, so oxygen will have a higher affinity to electrons. So it will accept electrons from hydrogen. I mean hydrogen plus, right? So that is what does these numbers mean? This positive, negative value, you have to know they indicate the affinity to electrons. And this is a very important equation. We say you have cell potential, right? So you must have two half reactions. One half is oxidation reaction, the other half is reduction reaction. So your cell potential for the overall reaction should equal to the cell potential for the half oxidation reaction, or half reduction reaction minus the half oxidation reaction, which here is the electron donor, here is the electron acceptor, right? So it's always working this way. And be careful for these values. It's always using reduction potential, which means half reduction reaction. These values, all these values are given as half reduction reaction. Okay. For example, this problem actually is also in your PowerPoint. If you're given two half reactions, so one reaction is okay, the ferric gain an electron is reduced to be cis ferrous. Another half is oxygen gets reduced to be water, right? So because here is only one electron, in order to make the electron balance, I have to put a half to make it become, to balance the reaction. Actually, the electron number is still not balanced, but the atom numbers are balanced. In order to balance the electron numbers, you should times 2 for the cis ferric and times 2 for the cis ferrous. But when we apply the cell potential equation, so you don't need to worry about the number of electrons. It has nothing to do with number of electrons, right? So only related with the two half reactions. So when you have this question, it asks you to calculate the cell potential. So you're given the cell potential for each half reaction. How can I find the cell potential for the overall reaction? First step, list the equation here. This equation won't be given, so you have to know it, right? Keep in your mind. So second, very important step, you need to differentiate which one is your electron acceptor, which one is your electron donor. So here it is. The molecule, or we say the element that has a most positive delta E, sorry, here should be E, no delta, has the most positive cell potential should always be your electron acceptor, because it should have a highest affinity to the electron, right? Like here, if you compare these two, oh, they're both positive, so this larger one should be the electron acceptor. So the smaller one should be an electron donor. That means you should use that means you should use 0.82 minus 0.22, right? So if one is positive, one is negative, apparently the positive one will be your electron acceptor. Negative one will be electron donor, okay? So that is for how to calculate the cell potential. Now let's see the problem exactly it has. Here, right? So it asks you, it didn't ask you to calculate cell potential actually. It asks you in which direction will electrons flow spontaneously if the species shown in the half reactions are combined. So I will say which one is reduced, which one is oxidized. Since we say oxygen is the electron acceptor, so that means electrons should flow from ferrous ion, ferrous is 2 positive charge, to oxygen. When ferrous lose electrons, it becomes more positive, right? Become ferric ion. So the electron flow should be from, let's say, iron 2 to oxygen. Okay. So another practice problem is here, the thermodynamic electron transport, still related with cell potential. If you're given these two half reactions and you are asked to calculate the cell potential E and the delta G, or maybe you're just asked to calculate delta G, but you still need to calculate cell potential first, right? So let's say cell potential, this half NAD is negative 3.32, oxygen positive 0.82. So which one will be your electron acceptor? Which one will be electron donor? The more positive one will be electron acceptor. So which means it should be oxygen. And when you calculate your cell potential, it's always acceptor minus donor. So 0.82 minus negative 0.32. Okay, so here is the negative, we still keep it negative. So that is your cell potential. So further use the equation we have for delta G equal to negative NF delta E to calculate the delta G. Okay, so that is how to use cell potential to calculate delta E, to calculate delta G. So the F is the Faraday's constant. Again, you know, in your exam, this number will be given if you need it. Okay. Here is a detailed calculation. Too slow, sorry. Anyway, that is a detailed calculation. You can check the PowerPoint. So that actually is the important part we discussed today. So how to calculate ATP, total ATP number, and how to calculate the cell potential. Okay. So next time we probably do a little bit more practice, and then we will continue talking about the oxidative phosphorylation.